Okay, so we had some sort of a introduction uh, sections for the last class, and then we will talk about the real process of the lithography. So, so say first of all, we start from the photolithography. So basically, this photolithography is basically how to make the semiconductor. So, so if you decided to learn about this photolithography in very detail, so you have to end up with about how to make a semiconductor devices actually. So this class is actually some chemical biology class. So we have some boundary for explaining the photolithography about some biochemical photolithography, such as the example that I gave you in the last class. So then, in terms, there you have some special the term explaining some biochemical or biological lithography. So we call it soft. So you can say photo, or generally speaking, soft lithography. So you can Google this soft lithography. So you will find that how to make the process that making the semiconductor devices to make how to connect it between this technique, which is lithography, to some or logical questions to answer biological questions, also to study bioprocess, which is the previous introduction is how the lithography, especially soft lithography, can answer your questions about how you can control the shape of the cell, and then the cells that adhere to the protein, and then how you control this protein morphology on the surfaces. Then you can control morphology of the cell, and then you can actually decide it or predict cell death or cell living. So that is some sort of system fundamental questions that how we can connect it between this lithography and this biology. Okay, so we will start about, say, I will change this title to the soft lithography, and then we talk about some soft photolithography that say. So generally, you can have some silicon surfaces, silicon wafers or glass, and anything that is very flat. So that can be a candidate for this starting materials for this soft lithography process. And then, say if you have some materials, generally a polymer, the polymers that are very sensitive, that are sensitive for an incoming photon. So the polymers that are very sensitive for the incoming photons. The sensitive means the gradations or polymerizations for the polymerizations. In other words, that if you're the sensitive means for the incoming photon is that you have some polymer materials, and then these polymer materials you have a unit of the monomer that is connected to each other, and the light is coming, and then they degrade. So the molecular weight getting smaller and smaller, which is this case. It's sensitive for the photon resulting to degradation of a given polymer. That is one sensitivity factor. Maybe if you have completely different polymers that are relatively short molecular weight at the beginning, and then the light is coming, and then this relatively short polymer is polymerized because of the energy that utilized is incoming to the solution. And then they actually attach to each other. Then and then this is further polymerization that I mentioned. So that can be also the sensitive to the photon. So you can say one is degradation pathway, the other one is relatively short, and then they make bigger and bigger upon the illuminations of the light. You can say those situations are sensitive for incoming photon. So I mentioned this. Then if you understand this part, say, let's begin with the case number 1, for example, degradation case. Okay. So the case number 1 is blue. So if you can, this degradation, possibly degrading polymers, you coat it here. Suppose you can coat it on surfaces. That can be a nanometer scale, that can be micrometer scale, depending on the viscosity of the solution. If it is too thick, then you rotate it by this system, then you have a thick coating. If this polymer solution is relatively low viscous, such as nearly similar to water, then you rotate the system, then you have very thin layers of the polymer. So you can control this thickness by the rotating per minute, RPM, and viscosity of a given polymer. Right? So you have a polymer, so a given polymer, this viscosity function, and also the RPM that rotating this whole system. Okay, so you can control this thickness from nanometer or micrometer. Okay, so then I'll delete this, by the way. And then the next step is if you have this some mask. So mask is a real mask. The mask means that you protect the illumination. Right? Protect illumination means you have some, say, I can draw like this way, and you have polymer solutions as spin coated. And then you bring a mask that maybe looks like this. Say, for example, like this. So say you can actually imagine the situations. So you have some surfaces and you spin coated polymer. Then you have some mask. That mask has a line. There's a line here and here and here. So you can mask this. So through this, photons cannot penetrate. So other area without this mask, you can actually light is coming to the polymer solution. So that's why I draw like this way. Okay. And then this system is entirely very thin. So you can imagine this the silicon wafers which is making for the semiconductor. So it's very thin device. Also, this polymer is in the micrometer, nanometer scales is very thin. Also, this mask is also very thin. So everything is very thin. So you have to understand these situations. So then you bring this to the some illuminator that has a light source. Then what happens is here that this mask is protecting the photon is coming. So that you have then you have a mask. Say I'll draw it similarly like this way. So actually this mask is physically contacting this polymer solution actually. But I draw this way. And then this illuminator light you provide. This is the difference. So what happens is this area is A, which is no exposure to photon, right? But this area B is exposed, right? This is exposed to the photon. So if you remember this part that is sensitive, then the case number 1 is degradation, right? That means if you zoom in this area, then these polymers are now start degrading. But this area, not. This is maintaining the original molecular weight. So there is a difference. So you can say, what if the solubility of the degraded polymer, I should say in this case fragment, this degraded fragments, which is I mentioned this part, and the solubility of non-degraded part, which is original this part, which is not exposed to the light, are different. So this is a very, very important part, because now you have, say, the solubility against, for example, say you have acetone, for example, or ethyl alcohol, or dioxane, whatever solvent that you know it. There's a difference. Then for example, this degraded fragment soluble to acetone, but non-degraded polymer is not soluble acetone, for example, the situations. Or differently, degraded part solubilized in ethanol, non-degraded polymer not solubilized in ethanol. If that is the situation. So what if? So as I mentioned, what if? Then, say if this happens in acetone, for example, we imagine these situations. So you put acetone here. Then what happens is, you already predict, but I should draw situations now. These parts are gone because they solubilize in acetone, so they will be gone. And then only the remaining part will be like this, because we have the photomask is existing here, right? And this photomask, and this is the remaining part, and this is the remaining part, right? And if this is a physical devices, because I mentioned that to you that this is everything is a thin device, and that there's a mask here, so you can physically remove this mask, right? So then you remove the mask, and then you remove this part, then you have that if this is silicon wafer, then silicon is exposed, it's not exposed, and non-exposed. So this is a key part. Then how we make a pattern on surfaces. This is a very, very general principle how we make actually the lithography circuit in the electronic semiconductor industry. But we as I mentioned, that we focus on this biological soft lithography. So I'll bring this part in here again. So that I will delete everything. So I'll bring this part in here again. And now we have the glass or silicon wafer or glass that has the polymers are remaining on 3 positions. So we start from here. Okay. And then what if we have some rubber like elastomer, so something like you can it's a rubber. But as you know, the rubber can be a solution state in a certain process where the rubber can be solidified. So actually you can call this rubber. But there is a rubber you actually before to make a rubber that you imagine, then there's solution part exists. Such as we have a rubber band, for example. When you make a rubber band, that previously was actually solution, and then you have a curing process, and then it becomes a solidified to actually you can handle this rubber band before the actual solution. So you can elastomer solution. You pour. I'll do this. Say, and then we've got a 3-part, 1, 2, 3, and then I'll use a red color to express the rubber-like solution. So you can have you pour the rubber-like solutions here, and then you cure. So you can give a curing process. And then because the solution to solid, this is a curing process. This is curing part. You cure to make a solid, and then you put this out. You put this lift off, and then another part is lift off. The elastomer. Then what you end up with is this one, the red one that has say 1, 2, 3. That looks like this. And this area is very flat because everything is. So when we start from the silicon or glasses, and I mentioned everything flat is possible a candidate for this process, right? So this, since the elastomer solutions are poured to the system, and then you lift off for the red part, and then this can be a rubber, which is very flat in the extruding part, right? This is the makes sense. So you can say. This can be, looks like this is rubber, so elastomer, so it can be a stamp, look like a stamp. Look like a roll as a stamp. So you can make a stamp out of it, right? Say, if your situation's here, for example, the next time, or next step, you have, say, collagen protein, or gelatin, or laminin, or other proteins. For example, this is extracellular matrix protein. Solutions here. So you can solutions here. Then you bring this stamp to touch the solution. Okay, so you touch the solution, and then you got a stamp now. The stamp has the protein solution. For example, can be a collagen or mixtures or gelatins or laminin, whatever to culture the mammalian cell, or it can be a serum. So remember that the previous part, when if you forgot that, you go back to the mammalian cell culture part, and then there's a serum protein that I explained. So it's a serum solution. Then the serum solutions are adhere to here. The solution can touch some soaking solutions, and then finally, say you have a glass, and you got a stamping. Okay. The solutions can be, you got a physical stamp like onto the flat surfaces, and then what you have to do is, say you got a 1, 2, 3, 4, and actually is this line will be the line. The 4 lines of the protein or serum can be formed in a very controlled way. They can be so these dimensions, the size or dimensions, or depending upon, can be controlled to initially what mask. But the photo mask, I deleted the photo mask part. The photo mask was here initially. The size depending on what the photo mask you design it. So that is small, such as micron or nanometers, then there can be a line of nanometers, a line of micron. So the lithography is basically in soft lithography, the scale of these patterns are usually about 100 nanometers, about to say 100 microns. A few 100 nanometers, or can go down, can go up, but typically speaking, you have a scales of line. You may have questions about how the line size that it can be a 1 micron, that can be a 10 micron, it can be 100 nanometers, or such as this scale. Because the size of the cell that I mentioned before was about more than 10 micrometers in a diameter. So that's why this is suitable for this scale. About this scale is suitable appropriate for the cell dimension scale and some other else. Okay, this is one general, the typical process of the soft lithography part. I will do the second one. This is one way. So you can basically, this approach is you can have basically you make a stamp. So you can make a stamp here. And the second one is actually you use this stamp by itself. Say the stamp that was the case number 1, and then use this stamp as a stamp. So stamp means actually this device itself, the elastomeric device is actually to deliver biological molecules to the certain surfaces. This is kind of carrier system. So this is the stamp means a carrier of the molecule that you actually want. So the molecule is just final the things that you want to have, but not actually stamp is by itself is useless. But the case number 2 is actually the stamp itself is a final product. So stamp itself is final product. This is different case. Okay, so that is, say you have this stamp, for example, and make the stamp upside down. So previously I make upside down, but now it's a right way. So this we use it this as a final product because some cases are, say, you have a neuron, for example. So this is a neuron cell and this is dendrite, and there's other neuron cells here. And the dendrite, so there's a network out of it. The cells responsible for this cell can live here, and dendrite goes here. Cell dendrite goes here. So you can control the surface morphology of. This is one of the example. One of the examples that how you can control the cell morphology, especially neurons or stem cells or muscle cells. Many other cells actually they can control how they questions that how you cells are responsible. Response for this different morphology of the structures. Because you have such as bone. Bone is not completely 100% filled solid. They're actually porous materials, right? The bone is porous materials. Actually, you can have so porosity is increased, and then the osteoporosity is actually disease. But actually, everyone else is not 100% filled. Is in a bone. But the bone cells actually they can feel the overall morphology inside the bone. There's porous materials, so they can actually see the cells are able to distinguish between this how they rough or how they smooth and how they look. This is basically the biological process inside the body. So this is the kind of questions that how you can control this. You can make a for example like this, very long. Or some cases very sharp, or some cases you have something like looks like this. You can make some detailed photolithography technique you learn. Then you can actually make some various shape of this stamp. Okay, so we will continue this soft lithography for the next class. Okay, thank you very much.